Hey guys, it's Mrs. Esser, and today we are going to talk about cells. So, to begin, I want to talk about different cell scientists that I want you guys to know. So, our first guy is Robert Hooke. He was in charge of naming the cells. So, he looked at a cork under a microscope and it reminded him of the cells monks used to live in. Therefore, he named the cell because it reminded them of monk bedrooms. Our second scientist is Anton van Leeuwenhoek. He was our first scientist to view living organisms under a microscope. So he looked at scrapings on his teeth, pond water, all different types of organisms. Robert Brown discovered and named the nucleus. And then lastly, we have Matthias Schleiden, who was a botanist, Theodor Schwann, who was a zoologist, and Rudolf Virchow, who are the co-founders of the cell theory. So what is the cell theory? So there's three different parts. First, stating every organism is composed of one or more cells. So one cell would be considered unicellular, more cells would be considered a multicellular organism. Two, cells are the smallest unit of life, meaning that small cells will still follow Stirninger, meaning they synthesize, transport, excrete, regulate nutrition, growth and development, respiration, reproduction. And then the third step, the cell theory, is that all cells come from pre existing cells. So that's something we're going to learn more about in mitosis and how cells divide. So, first, I want to start off by comparing two different types of cells. So, we have prokaryotic cells and eukaryotic cells. So, breaking apart our root words, we know pro means before and kary means nucleus. So, a prokaryotic cell literally is before the nucleus existed. So, looking at our picture, we can see prokaryotic cells are going to be smaller unicellular organisms. They don't have a lot going on. They lack membrane bound organelles. Eukaryotes, I remember you are a eukaryote. So U means true, kary meaning nucleus. So eukaryotes will have a true nucleus. So that means our DNA, our genetic information, is held inside the nucleus, and we still have all of those membrane bound organelles. We can see eukaryotic cells are a bit larger, more complex than the prokaryotic cell. So, prokaryotic cells will have our genetic material just free floating in the cytoplasm. So, a term I want you to become familiar with is a plasmid, which is circular DNA floating around that cytoplasm. And then we're also going to be looking at chromosomal DNA. We get more into this in our biotechnology unit, but I want you to know that plasmid, small circular DNA within our prokaryotic cells. Eukaryotic cells will always have a nucleus in order to separate genetic information. Some of the kingdoms that do that are Plantae, Animalia, Fungi, and Protista. So, just some things I want to point out between prokaryotes and eukaryotes and similarities they have is every single cell is going to have a cell membrane. There has to be something there regulating intake and outtake of items, helping the cell maintain homeostasis and just acting as that barrier inside and outside the cell. Every cell will also have ribosomes because we know proteins are essential to life. So every living thing has to be able to do protein synthesis. Therefore, you need ribosomes since they are the location where proteins are made. The cytoplasm is that gel background of the cell that prokaryotes and eukaryotes will have. And then we also need DNA or some genetic information in one form or another. So I say that because prokaryotes have plasmids, where eukaryotes we're going to have our DNA either in the shape of chromatin or chromosomes. So let's look at all the organelles I want you guys to know. So our first one, the nucleus, which hopefully we've heard about before. So that's going to contain our cell's DNA in our eukaryotic cells. So our DNA codes for genetic instructions for making proteins. We know a small segment of DNA is called a gene that can turn on or off to activate new proteins to be formed. Other items to know about the nucleus is around the nucleus, we have our nuclear membrane, which kind of is like a cell membrane. It regulates what comes in and outside the nucleus. And it's the nuclear membrane's reason that the DNA can't leave because remember, the double helix is too big, but it does have these small nuclear pores where mRNA, since it's single stranded, can fit out. Within the nucleus, we can see this nucleolus literally means small nucleus. So it's a small dense region on the inside of the nucleus that is in charge of making ribosomes. So the ribosomes will also fit outside the nuclear pores and they'll go to something called the rough endoplasmic reticulum that we're going to talk about later in this video. Another item and new word is chromatin. So that's just the state your DNA is in during resting phases in the cell. So chromatin, I kind of think about spaghetti. So it's your DNA bound to these proteins called histones. It's kind of just hanging out before division occurs. Our cytoplasm, cyto meaning cell, plasm, gel, liquid. So the cytoplasm is the gel, liquid, or background of the cell. So that's what's going to be in charge of holding all of the organelles. So you're going to hear this term organelle a lot throughout this unit, which literally means mini organ. If you think about your organs within your body, it's helping your body function. So organelles are small organs inside the cell that allows the cell to operate as a whole. Ribosomes, we should know this from protein synthesis. So we have transcription occurring in the nucleus, and then the mRNA travels to the ribosome for translation. So the ribosome is where the protein will actually be made. So we know proteins are made at the ribosome, and we know the ribosome is made out of rRNA or ribosomal RNA. So we can find the ribosomes free floating in the cytoplasm, or we can also see them attached to the rough endoplasmic reticulum, which you can see right here. So we talked about the ribosomes are made within the nucleolus. They leave that nuclear pore and go to the rough ER. So here's our smooth and our rough ER. So smooth, we can look at this picture and the line actually looks smooth. So there's no dots on it like the rough endoplasmic reticulum. So smooth does not have ribosomes, and it's going to help make lipids. We can think about phospholipids that are made or used in the cell membrane. So lipids are an essential part of the cell. And then rough endoplasmic reticulum, I always think if I were to be able to feel this picture, I'd feel all those blue dots and it would feel rough on my fingertips. So rough endoplasmic reticulum is in charge of transporting those ribosomes that were made in the nucleolus and transporting them out into the cytoplasm. And our ribosomes are where protein synthesis is made. So sometimes people will say the rough ER has a role in protein synthesis. Our Golgi apparatus, you could also hear it Golgi complex, Golgi body, all the same thing, tomato, tomato. So usually you'll see the Golgi apparatus near the cell membrane because it's kind of the mailman for the cell. So it's going to package proteins, materials to be sent outside the cell, and then it will push that out of the cell membrane. So our Golgi apparatus, much like the mailman of the cell. Lysosomes, we've also heard them as the suicide squad of the cell. So there's sometimes when cells are no longer useful for our bodies, so the lysosomes will actually release enzymes in order to break down that cell. Or perhaps a small organelle isn't working well, the lysosome will come break that down. It can break down lipids, carbs, proteins to be recycled as well. So you can think of lysosomes as a Lysol spray you're spraying to clean. So lysosomes are helping recycle and release waste and things we no longer need in our cell. Vacuoles, these we'll only see in our plant cells or in our protista cells. So these are used to store water and nutrients. Actually, I'm false saying that. There's some animal cells that have vacuoles, but if they do, they have them very small. So vacuoles are storage units. We can see them a lot in plant cells. When it rains, that vacuole will fill up with water, and then as the cell needs it, it will release water from the vacuole and shrink again. So it's kind of storage for a later time. The chloroplast, this is review from our bioenergetics unit. So this can be found in plants, protists, any autotroph. So this is the organelle that's going to capture the sunlight energy. Specifically, we know the thylakoid absorbs that sunlight in order to convert it into glucose, and then that glucose we can later use during cellular respiration. So chloroplast, the site of photosynthesis, the main goal is to create glucose, and oxygen is released as a byproduct. Our mitochondria, also going back to our cellular respiration. So the mitochondria is the site of aerobic respiration, creating 36 ATP. So we're taking that chemical energy from our glucose. We're breaking that glucose down in glycolysis, which we know is our anaerobic respiration. And then if we have oxygen, we can breathe in and go into aerobic respiration, which would occur in the mitochondria where the most energy is produced. The cytoskeleton of the cell, that's the structure that basically helps support the cell. If you think about your skeleton, helps give you shape. So the cytoskeleton is a network of protein filaments that helps the cell to maintain its shape and structure. Up next, we have our centrioles. So the main function of the centromere is to act in cell division for animal cells. So we're going to look at this in our mitosis. It's used in metaphase and anaphase to pull the genetic information from the middle of the cell to dividing into two separate cells. And last but not least, our cell membrane is our organelle, which we should know a lot about. We have our proteins that can be used in active or passive transport to move molecules through the cell membrane. We have our phospholipids with our phosphate head and our fatty acid tails. So, just a couple more things before we wrap up with the difference between a light microscope and electron microscope. So, light microscope is what we use at our high school level. It's going to allow us to see some of the organelles, see the cell wall, but the electron microscope is going to be a lot more detailed. So we can see the electron microscope here. I believe this is a picture of an ant, so it's very detailed, uses electrons to take images, and those cost about $50,000 at the cheapest, so it's a lot more detailed. If I ask you to find the total magnification, you always take the eyepiece, which in my classroom is always 10 times the objective lens. So if you're on low power, it's 4x, medium 10x, or high power 40. So if I was on low power, I'd take 10 times 4. My total magnification would be 40. And our last slide for today is this term called a stem cell, which we're going to get more into. But a stem cell is an undifferentiated cell that can give rise to different types of cells. So when you are born, the stem cell is the original cell. Those genes can differentiate and lead to creating neuron cells, muscle cells, red blood cells. But once a cell is differentiated, it can never go back to a stem cell. So that's it for our cell structure video.